I am John Fetter, a product manager in emerging chemical synthesis. Today I will be telling you about our DNA-encoded fragment library that can be used for drug discovery screening. This slide shows examples of our cutting-edge research products across the drug discovery workflow. Starting on the left, this DNA-encoded library, also referred to as DEL, is used during the early discovery phase when you're trying to find hits and develop them into lead compounds. We also offer Synthia software that can be used to design efficient synthesis reactions. Our photoreactors can help improve experimental reproducibility. We're offering protein degrader building blocks, and we also offer polymers and nanoparticles that can be used for drug delivery. With DNA-encoded library technology, each compound is attached to a different DNA sequence, which can also be referred to as a barcode. These barcodes can be used to identify the compounds. It is impressive that an entire library of labeled compounds fits in a single vial. The target of interest is attached to beads through a tag. Then the target and DNA-encoded compounds are incubated. Those compounds that have a higher affinity bind to the target. Those not bound or with lower affinity can be washed away. Then the DNA is amplified using PCR and analyzed using next-gen sequencing. The sequencing data is used to translate the DNA barcodes into the hit compounds. The Dynabind DNA-encoded library that we're offering is different from other DELs in a couple ways. First, the library contains small compounds referred to as fragments. Second, the DNA can hybridize and form dimers so that pairs of compounds can bind to targets. The library has 614 labeled fragments. Since pairs of any combination of two fragments can form, there are more than 370,000 combinations. There's also flexibility in how a compound pair can bind to a target. This way the library covers even more chemical space than 370,000 compounds. After you get hits, you can test various linkers with the compound pairs to optimize binding affinity. Single binders can also be detected. We are offering this DEL technology as a kit. In the past, DEL technology could be accessed through CRO agreements with milestone payments. It was also possible to set up an internal DEL screening program, but that could be a huge investment. This kit allows for testing without any agreement. In this kit are 5 identical vials. Each of these can be used for one experimental condition. For example, you could test your target in triplicate and include 2 negative controls. During analysis, the data associated with each vial will be used to generate a list of hits for that experimental condition. Here are more details on the library design. The rule of 3 from the Congreve paper was a guide during compound selection. Compounds were intentionally selected to be diverse. The average Tanimoto similarity score is low at 0.195, indicating diversity. Pains, pan-assay interference compounds were removed, and compounds known to be toxic were removed. Quality of the library is also better than that of a standard DEL because each DNA tag compound is HPLC purified and validated by mass spec. That is not possible when you have a billion compounds in a library. The results you get from the Dynabind screen are more reliable. On the product page on our website, we have 2 protocols. They can be found on the page under Documents. One is for the assay, and the other is for loading the next-gen sequencing data into our portal for analysis. The assay protocol has 2 options for detecting fragments. One is for using a his-tag protein with magnetic beads, and the other is for using a biotin tag protein with streptavidin-cephurose beads. The protocol also has details for the PCR amplification of the barcodes, and then for the next-gen sequencing. The left side of the slide shows the data input form on the portal. Once you upload the next-gen sequencing data, software will analyze the hits by detecting the fragment barcodes. It will provide the top 20 for each of 5' single hits, 3' single hits, and paired hits. On the right, you can see an example of the output, where we give the actual structures. There's no additional charge to use the software to see the structures. It is a good idea to run the protocol to detect the single and paired hits. You will likely see overlap between the 5' and 3' hits. Weaker binders may not overlap since the DNA tags can influence them. Typically, some of the single hits will show up in the pairs, but not all of the pairs will show up in the single hits. After you get your hits, there are a few options. If you want a quick validation of the hits, you can work with Dynabind and they can determine the kinetics with the DNA tag fragments such as on rates, off rates, and KD. They can also do affinity maturation, in which one fragment is held constant and the other is varied. If you want to do your own validation, you can order the fragments, and most of them will be available in our catalog. With the fragment pairs, you can test various linkers, or you can use Dynabind's services to help develop linkers. How do you buy the kit? You can go directly to the product detail page by typing in the web address sigmaaldrich.com/del, or if you're already on our website, you can search for Dynabind or the catalog number Dyna001. The list price is $12,540. If you would like to learn more about DEL screening, in 2019 we had a special issue of the Acta containing 2 reviews on DEL technology. One review gives more background on the development of the fragment DEL screening library. The second review discusses different types of chemical reactions that are compatible with DNA that can be used to create DELs. If you have any questions about buying the kit, you can contact me or Ben Glasspool, who is head of our group. Thank you for listening.